Hello, this is Inhibitory Effects of Allogliptin and Pyridin on MMPs potentially preserves blood-brain barrier integrity following traumatic brain injury and ischemic stroke, a molecular docking study. I am Amol Balakrishnan. This is my email. I attend school in Marietta, Georgia, specifically Lassiter High School. The primary main focus is how can computational methods, specifically molecular docking, be used to identify therapeutic drug candidates that alleviate symptoms of traumatic brain injury and ischemic stroke. For our cases, TBIs are just, most of us know what they are. They're just injuries that affect brain function. They usually occur when external forces strike the head. They affect 69 million individuals per year, which make them pretty significant. Ischemic strokes, on the other hand, are less familiar, but they're just when a blood vessel is obstructed, usually by a blood clot, preventing blood and therefore oxygen from reaching the brain tissue. 87% of all strokes are ischemic. These conditions can be pretty fatal, and they often lead to long-term disability. Now, here's just a slide that depicts the TBI and ischemic stroke in detail. So here is where the TBI would be, and here is the blood clot preventing blood from entering, and therefore oxygen from entering the brain's tissue. Now let's give you some introductions on the protein. This would be the receptor in our molecular docking study. The protein group that we would primarily focus on are matrix metalloproteinases, abbreviated as MMPs. They are zinc-dependent proteolytic enzymes, so that means they break down proteins, that play a significant role in embryonic development, the formation of blood vessels, and general cell repair and remodeling. Now this is important to consider. MMP levels are low during normal brain conditions. Now, let's take a look at MMP9, gelatinase B, usually it's sometimes called. So this is a part of the natural response to the injury. MMP levels increase following TBI or stroke, and this degrades the tight junction proteins of the blood-brain barrier, essentially rendering it useless. Now, what the blood-brain barrier is that it's the protective layer of the brain that lets certain things in and out of the brain region. This results in brain swelling and enhanced neuroinflammation. This is our ligands, or our substances that will bind to our receptors. First, we have allogliptin, which is depicted at the top. It's an FDA-approved drug used to treat diabetes by ultimately increasing insulin in the body. It has known MMP9 inhibitor properties, which can be seen through mice mRNA expression. But if it directly binds to MMP9, which can be seen through molecular docking, is not clearly known and has not been done. On the bottom, we have pyridin. It's a natural anthraquinone, and it's basically an organic compound found in plants and fungi, known to have anti-inflammation and anti-cancer properties. Now, pyridin's interaction with MMP9 is not fully known, and its molecular docking studies, including the substance and MMP9, have not been done before. So the bottom line is, at least for novelty, neither allogliptin nor pyridin have previously been studied for the specific molecular interactions with MMP9 using molecular docking, and their binding mechanisms remain to be fully elucidated. So, with these introductory concepts, it's pretty easy to figure out the big idea, which is to evaluate the potential of allogliptin and pyridin to protect the blood-brain barrier, which is the BBB, following TBI or stroke by inhibiting MMP9. This is done by using molecular docking to assess their interactions and therefore therapeutic potential. Now, let's move on to our methods, in silico modeling. So molecular docking was carried out with Autodoc Vena. It's an open source program provided by Scripps Research that's obviously used to simulate protein and ligand docking. It predicts binding orientations and affinities of molecules to a specific receptor/target within an area that we can choose. In addition to Autodoc Vena, we have Schrödinger glide, which is used to substantiate the results of the interactions. Obviously, since they're two different programs, they're not going to have the same exact results. But what glide is that it's an industry-leading solution for molecular docking that's similar to Autodoc Vena, determines the specific best fit interactions between ligands and receptors. And this best fit means that it has the least energy used within a predetermined area that, again, we can choose. So now let's move on to our structures. How did we attain them? Well, for our receptor, we had the catalytic domain of MMP9. Obviously, we're not going to have the full protein. This is structure ID 4XCT, which can be seen here. There's actually a ligand in this representation that we actually had to remove prior to molecular docking. Now, importantly, it has a resolution of 1.3 angstroms, below 2 angstroms, indicating a high resolution and accurate atomic structure. Now, for our ligand structures, we obtained these using PubChem, specifically allogliptin and pyridin. The IDs can be seen right here, and their models can also be seen right here. We obtained these using the SDF format. Now, before we can begin docking, protein and ligand preparation must be carried out. For Autodoc, this can be done through Python scripts, which can also be done automatically. It consists of, in general protein preparation, consists of assigning charges, usually Coleman, adding polar hydrogens, and performing the appropriate file conversion, which is .pdv to .pdbqt, Q meaning charge, and T meaning atom type. The similar steps are done with glide for protein preparation, but they use different what is called modules. A very similar task is done with ligands so they can be optimized and recognized by the Autodoc software. The same is done with glide, but through, again, modules. This is an image depicting of what a protein preparation step looks like, but it's not done in Autodoc, but it's very similar. Now this is for docking. So it's important to realize that MMP9 requires a zinc ion as a cofactor. Again, one is used for catalytic activity, while the other one is used for a structural role and stability. So when determining the coordinates of the binding site of what allogliptin and pyridin would bind MMP9 to, we had to keep this constraint in mind. This is done manually in Autodoc Vena and using a module within glide. Then we'd had to determine the exhaustiveness, or the computational effort of the docking, which is a value which we set as 32 before docking in Autodoc Vena. And within glide, we use the XP docking strategy, which just means extra precision before docking. Now, for visualization. For Autodoc, we use open source Amol. It is used to visualize and help analyze docking results fully. It's a user-sponsored system, meaning open source, and it was initially created by the scientists, but now it's maintained and distributed by Schrödinger. Now, after glide docking, visualization can just be done within its own glide software. This is typically what what Amol would look like, and this is just the logo. For molecular visualization results, pause here. These were modeled using Amol. These are not glide figures. For molecular visualization results, pause here. These were modeled using PyMol. These are not glide figures. So for discussion, a negative k-mol binding affinity basically just means that autodock and both glide have performed docking successfully. The ligand's presence in close distance to a zinc ion and other donor atoms suggest strongly that a metal coordination bond was between them, but the ligand and interact with the zinc. So because ligands formed polar interactions with critical amino acid sequences, along with almost certainly chelating with, which just means like interacting with the catalytic zinc ion, allogliptin and pyridine inhibit MMP9 activity very probably on a direct level that has not been seen before. With this, again, it's very probable that MMP9 inhibition caused by the interaction between this enzyme and two ligands can therefore help preserve BBB integrity following TBI and stroke. Now, in conclusion, basically what we said here was set into discussion. With this, it's our expectation that this study will help confirm previous findings and lead to potential future experimental clinical research and trials. The previous findings, meaning like those with the mice and mRNA expression, aimed at restoring BBB integrity by therefore alleviating symptoms of patients with traumatic brain injury and stroke. Citations are here. Thank you for listening.